Type 2 diabetes has become a raging epidemic, with about 1.4 million new cases diagnosed every year. In order to put an end to this epidemic, researchers are trying to understand the molecular pathophysiology of this intimidating illness. Normally, when there are elevated levels of glucose in the bloodstream, like after a meal, the pancreas releases insulin. Insulin then binds to a membrane protein of a cell and causes a series of reactions that induce glucose transporters to move into the membranes and facilitate the movement of glucose into the cell. But type 2 diabetes is associated with insulin resistance. This means that target cells show a decreased response to insulin. In other words, someone is insulin resistant if they need a lot of insulin to be released in order to get a certain amount of glucose into the cell. And essentially, because the body is not able to transport glucose into the cell effectively, these cells starve. Recently, it's been proposed that tyrosine phosphatase inhibitors may be a viable treatment for type 2 diabetes. But what's a tyrosine phosphatase? Phosphatases are enzymes that remove phosphate groups from a protein. A tyrosine phosphatase removes phosphate groups from phosphorylated tyrosine residues on proteins. Phosphorylation of tyrosine residues on proteins is a common post-translational modification, and whether the phosphate groups stay on a particular tyrosine residue on a protein or are removed by a tyrosine phosphatase can have a dramatic effect on a protein's function. We're interested in insulin receptor membrane proteins. Insulin receptors consist of two subunits, a ligand-binding subunit and a tyrosine kinase subunit, which is responsible for phosphorylating certain tyrosine residues. Signal propagation happens in a very specific series of steps. First, when insulin binds to the receptor, the tyrosine kinase subunit is activated after autophosphorylation of crucial tyrosine residues. Then, the tyrosine kinase phosphorylates other insulin receptor substrate proteins downstream. This then enables propagation of the insulin signal, which eventually leads to letting glucose within the cell. Now, the opposite of tyrosine kinase is a tyrosine phosphatase, which removes phosphate groups from crucial tyrosine residues. Tyrosine kinases and phosphatases work together in regulating signaling within the cell. While kinases are critical in controlling the amplitude of a cellular response, it's widely believed that phosphatases have an important role in controlling the rate and duration of these responses. This is especially important for diabetes research, since the rate and duration of which an insulin receptor stays activated has a huge impact on the propensity for glucose to enter the cells. Essentially, the longer the signal transduction pathway stays open, the greater the chance for glucose to enter. In order to figure out what kind of effects the tyrosine phosphatases have on the insulin signal transduction pathway, researchers have turned to tyrosine phosphatase inhibitors. So how does this work? By placing a small molecule inhibitor, tyrosine phosphatase, which would normally turn off the insulin signal transduction pathway by removing phosphate groups from key tyrosine residues, will be physically prevented from doing so. This results in the insulin signal transduction pathway to be active for longer periods of time. One popularly utilized inhibitor type is the uncompetitive inhibitor, which binds to the enzyme substrate complex. In this case, the enzyme is the tyrosine phosphatase, while the substrate is the key tyrosine residues. The inhibitor binds to the complex formed and prevents product formation, or dephosphorylation in this case. To demonstrate the effects of reduced levels of tyrosine phosphatase activity, researchers utilized mouse models and introduced inhibitors in their experiments. By using an inhibitor, some researchers were even able to completely delete tyrosine phosphatase activity. So what were the results? Without the inhibitor, researchers saw that in diabetic mice, tyrosine phosphatases would proceed to dephosphorylate the key tyrosine residues on the insulin membrane proteins, which would mean that the insulin signal transduction pathway would cease. Hence, glucose transporters were not recruited and activated, thus limiting the amount of glucose that could enter. In other words, these cells were insulin resistant. Even with insulin present, the cells were not able to bring in glucose since the pathway had been turned off. But with the addition of the inhibitor, tyrosine phosphatase activity was deleted, and this meant that the tyrosine residues remained phosphorylated. The insulin signal transduction pathway continued to activate glucose transporters, and glucose continued to enter the cell. Researchers determined that this, in turn, increases insulin sensitivity. Being insulin sensitive means that you only need a small amount of insulin to be secreted in order to get a certain amount of glucose within the cell. But what were the signs for this conclusion? After locking up phosphatase activity, the researchers saw a greater likelihood for glucose to enter the cell. They also found decreases in plasma glucose and decreases in overall insulin levels, which suggests increased insulin sensitivity, since less blood glucose implies more cellular glucose, and it took less insulin to get this result. This deletion of phosphatase activity enhanced insulin-induced insulin receptor signaling, suggesting that phosphatases act by suppressing insulin receptor activation, which is a key step in bringing glucose into the cell. Basically, getting rid of tyrosine phosphatases can help fight insulin resistance. Because the biggest issue with type 2 diabetes is insulin resistance, and decreasing tyrosine phosphatase activity has shown increases in insulin sensitivity, could this mean that we're at the preliminary stages of curing type 2 diabetes? In order to reduce the diabetes epidemic, researchers are engaging in novel projects targeting the molecular basis of the illness. With more research and some more time, it surely seems the results are quite promising.